Hello, my name is Peter Schneider. I am with a group of world experts on peripheral vascular disease and the effects of paclitaxel and its role in the patients that we see on a day-by-day basis. And I would, in this next few minutes, we're going to have a kind of a round-robin discussion on what's the latest information and where we've been up until now. So with that, would the panel please introduce themselves?
Hi, my name is Eric Secemsky. I'm an interventional cardiologist in Boston, Massachusetts.
Hi, my name is Christian Behrendt. I'm a vascular surgeon in Hamburg, Germany.
And hello, I'm Konstantinos Katsanos. I'm an interventional radiologist from Patras, Greece.
Very good. And with that, we'll dive in. So as everyone who follows the peripheral vascular space knows, the use of paclitaxel and its potential both positive and negative effects have been the subject of intensive research. And what we'd like to do first is cover the research that's been done to date. And probably one of the most important articles was published initially by Dr. Katsanos back at the end of 2018. So Konstantinos, maybe you could just give us a minute on how you came up with this concern, the potential that there could be a mortality signal, and kind of your view of the research that you did initially.
Thank you very much, Peter. So basically, I think there is no question that a number of randomized studies have shown that paclitaxel is a very effective anti-restenotic agent and can consistently reduce target region revascularization in a number of studies and with a number of different devices. So back in mid-late 2018, we noted that there was a trend towards a higher number of all-cause deaths in a number of studies, and we decided to pull this kind of data, and we published our first meta-analysis end of 2018 that highlighted our concern with this increased medium and mid-term and long-term risk of all-cause death, basically.
Very good. And of course, a tremendous amount of follow-up research was done after that initial signal was identified in the randomized trials. And we just happen to have two of the top researchers in the world, Dr. Secemsky and Dr. Behrendt. So let's go to you first, Eric. Most of your work is in the US. Dr. Behrendt will discuss work done in the EU. Eric, could you just give us a summary of your research to date and others that you think is important?
Yeah, so I think that, Costa raised an important concern in our field is we never want to do harm, and we didn't have a lot of consistent long-term data for these paclitaxel devices. So I think it was important for the community to generate as much evidence supporting or refuting a mortality signal as can be done when practice had already really incorporated drug-coated devices pretty permissively into vascular intervention. And so, some of the earliest stuff actually came from Peter and others who looked at device-based trials and tried to get an understanding of at a specific device, because we know many devices went into the meta-analysis. Do the same mortality signals exist, and is a relationship between dosing? And so we saw early on some individual programs for different drug-coated balloon companies and stent programs that didn't necessarily reproduce the signal within their trial data, but again, with limited comparison, small control groups, and having to kind of amass data, both observational and trial data, to get powers to look at this. And so we've kind of expanded how we've been able to glean the evidence that do exist already to better understand the signal. We have looked at a randomized control trial that was ongoing called the Voyager PAD trial, which didn't randomize to a drug-coated device versus a non-drug-coated device, but it did study this as part of the trial and looked specifically at endpoints comparing drug-coated devices and non-drug-coated devices and those treated with endovascular treatment. And there was no mortality difference in this study showing between the non-coated and uncoated devices. We've also seen real-world evidence play a very large role, and our group, Boston, was a primary driver of using Medicare claims data to ascertain long-term survival in patients treated with either drug-coated devices or not. That was called the SAFE PAD study. It was designed with the FDA, and there's been a number of updates for this study that has now exceeded follow-up of 6.5 years after an index treatment, and has failed to show in more than 150,000 patients a difference in mortality among those treated with drug-coated devices and not. There's also been important registry studies from the VQI, from the NCDR PVI registry, that have also not supported the primary results seen in the meta-analysis, but again, have suffered to some degree about getting the full 5 years of follow-up. Many of these have median lengths of follow-up of 2 to 4 years. So we've seen individual trial program, real-world evidence using claims data and registry data, all with strengths and limitations, but none reproducing this signal. And I'll hand it over to Christian maybe to talk a little bit about his research, and then the one randomized trial that was conducted that's outside the US called SweetPad.
Yes, thank you, Eric. Yeah, back in 2018, when this debate started, we were more or less impressed by the work Eric already did with Medicare data, and we tried to confirm or reject the signal first in health insurance claims data from Germany. As you know, we have the advantage of a very early market access of coded devices in Europe. So we used health insurance claims data from Germany using the Barmer Health Insurance Fund. And this is very rich data, and we tried to dive into that topic above the knee, below the knee, using the OPS codes for drug-coated balloons and stents in PAD treatment. And as Eric already introduced, we were also not able to confirm a safety signal, neither for overall mortality nor for amputation rate or cardiovascular event rate. So at the end, we ended up with completely opposite results, and this made us thinking about the possible reasons for these opposite findings. And from that on, we tried to dive into the differences and possible selection bias in different data. And I have no doubt that Dr. Katsanos found something in the data, but it was simply not possible to confirm with real-world data from Germany. Because you raised or you introduced the SweetPad, what is an excellent sub-analysis of the Swedish pragmatic trial. I mean, this is a little bit different from what we did with real-world evidence. You know, the SweetPad had a design and was powered for certain endpoints, but they also did this secondary analysis, and they were also not able to confirm any findings. So I think right now in Europe, at least to my understanding, there is no large observational data validly confirming a safety signal. But what we were able to describe is that the cohorts enrolled in randomized controlled trials, like Silver, PTX, and others, were not completely, but they were different from real-world data that we used from claims in different countries. So there are different proportions of women enrolled, there are different proportions of patients with diabetes, and so on and so on. I think this is the main or the most interesting aspect that we should discuss. How can we compare randomized controlled trials and generalize the findings to real-world evidence?
Yeah, and this is always a challenge, this how do you apply the results of a randomized trial to real world? And just, you know, this week the BEST trial came out, and that's probably going to be the main discussion around the BEST trial. Can it be applied? Can an RCT be applied? Nevertheless, with regards to paclitaxel, so many interesting things came out, I think, that are going to ultimately make our field better. The focus on mortality, what its causes are. Obviously, this is a huge issue for our patients. And I think up until now, we really haven't given it the attention and the focus that we probably could have. I think it'll be an important part of trials going forward. And, you know, beyond that, there are several things that we also looked for, and some of you alluded to it. So was this an association or a causal relationship, this signal that Konstantinos identified in the data? And so for that, we look at dose response. There has not, in my opinion, been any confirmation of a dose response We look for clustering of deaths. We have not found that, although I have to say, not all the trials have really divided the patients into subgroups enough so that whether we would identify a subgroup that was particularly vulnerable, although the work of both Eric and Christian have identified, say, claudicator versus critical, CLTI patients. And I think that's helpful. And then lastly, the consistency issue. Most of the signal from the RCTs was in the US-based trials, not in the Japanese or EU-based trials. And we really don't have a good explanation for that unless there were different kinds of biases that we had some hints to, but we haven't identified. Let me just ask next, what about new developments? Where do we go from here? Do we need longer follow-up in the real-world data? What about amputation risk, which has also been raised by Konstantinos, where I could imagine there may be a mechanism there with paclitaxel particulate, et cetera. Konstantinos, where do you think we should take this research next? What are you thinking about as ways to clarify whether this particular agent causes harm, or any agent, really?
Yeah. Thank you, Peter. I think I would like to touch a little bit upon the issue that Christian raised just previously about all the case mix differences between the randomized control trials and the real world data. And I mean, Eric has done amazing and sophisticated statistical analysis trying to eliminate confounding. At the end of the day, it's not about the real world data, it's also about the fact that the real world data is so far away from the patient characteristics that we actually studied in the studies. So for example, randomized studies, 90% were claudicants. In the real world data in the US, 50% of the patients suffered from chronic kidney disease, suffered from CLTI, etc. So completely different case mix of patients. And it really begs the question how we first translate randomized evidence and where do we actually put our value and our faith into the evidence that is being produced. Now, having said that, it appears we're going to be presenting an updated meta-analysis with, for example, at 2 years, around 5,500 patients. And it appears even when we include the 3D-pad claudication sub-study, we still have a significant result in terms of mortality for the case of paclitaxel at 2 years. Now, of course, I will agree that this seems numerically to be decreasing as more data becomes accumulated. And it begs the question, is it about, for example, that new generation devices with different formulation of paclitaxel may be safer, for example? And I have the feeling that those new devices might not suffer from the high rates of distal embolization like the first generation devices. And I would also put it to you, Peter, that we have seen the recent study of a new paclitaxel polymer eluting stent, with quite promising results, which is a different way of delivering paclitaxel. So I think there's no question that paclitaxel is effective. I have a strong feeling that it may be toxic as well, depending on the formulation and way of delivery. And this is where we have to come to terms to how to balance that.
Very interesting.
Make it.
Go ahead, Eric.
Maybe
please.
I'll just jump off that. I think those are. I think this area just is ripe for discovery in terms of how do we test these devices effectively without halting innovation and without reserving important technology for making it to our patients. I always one of the when I published a safe PD study in JAMA Internal Medicine, you're always a little bit anxious about the editorial. And, I wasn't sure what the editorial was going to hold. But the editorial for that paper, if you look it up, ended up talking about not the device controversy, but about the mortality risks associated with these patients. And it's just dramatic. Again, Dr. Katsanos made a great comment about this, that the trials don't represent necessarily our patients, and that's true. And so this all comes into what do our patients need? What are the risks we're willing to take how do we get to the risks, and how do we figure this out? I don't have a great answer for all of this. I think those are great concepts. I think that's great idea. Maybe there is something different about newer devices. I'm not sure, but I think as we move forward as a field, to me the big take homes, and Peter, you summarized this nicely, is how do we more consistently incorporate the importance of patient follow-up, medical adherence, long-term tracking of patients, and be able to recollect data. I mean, that is a no-brainer that we are always going to come out of this better than going into this whole controversy, because we've learned the importance of being able to identify those really important endpoints that can really put to rest or support these type of findings. And I think this will resonate with the next iteration of devices that are already being asked to look at survival, even if they're limus-based or different platforms, different polymers, to look at that long-term data. And I feel strongly also that if you're going to enroll a patient in the trial, this is the time to also, you have the money, you have the research team, the support, is everybody needs to be on maximal medical therapy, period, because that's what we can do in addition to tracking new devices. So there's a lot of extensions here. Where the research goes is hard. We're not going to run a 20 to 40,000 person randomized trial. We've talked a lot about this. So how we get to the absolute truth is still unclear, but I think that a lot of our data is really supportive of safety in a general real world population, not in the trial population.
And then Christian, what do you think? You've done some research on the amputation risk. Now we know from the randomized trials that the amputation risk is low in claudicators. But what about patients with CLTI? What's your current thinking on that?
Yeah, well, we have not published the data, but we were not able to confirm an excess rate of amputations in the CL, neither in the CLTI nor in the IC subgroup. And the data was large, so I mean we had enough events in that study, but there was no data on that. But please allow me to add one more comment to Eric's arguments, because I would strongly support this. We are discussing rather marginal differences here in terms of amputation rates and mortality rates. And this is quite interesting because we know from the data, and this is confirmed in randomized trials and in observational data, even in the best CLI and other trials, they don't take statins, they don't take the drugs, they just stop taking statins, stop taking antithrombotics. In Germany, 30% of the patients are just leaving the hospital without antithrombotics or even without statins. And this would save 30% of the legs and 30% of the lives. And I mean, as far as I remember from all the discussions back then with the FDA, it was very tough to get data on medical adherence and best medical treatment and all that. And I think this is something that we should dive into, because it may be more important to make sure they take the right drug or the right pill than the right balloon or stent. This is just something that gets into my mind all over during these discussions.
Yeah, that's a really, really great point. I mean, we're so enamored of our tools of the trade, but sometimes the basics aren't covered for these patients. And actually, that brings us to the last question we wanted to discuss. Where are the gaps? And so Christian just mentioned one of the major gaps, both in the trials and in our daily care of the patients. Maybe we could just briefly touch on, I'll just mention one of the gaps that we have is in the real world data, which I think is pretty accurate for mortality, which is terrific because that's a terrific use of the real world data. We don't always have a great idea of which patients have experienced paclitaxel in their lives and which ones are paclitaxel naive. And this is a confounding variable that we have with anything outside of a randomized trial. And in fact, many of the randomized trials didn't cover whether the patient had the other leg treated in the past, et cetera, with other devices. So this is a challenge for us. So what about other gaps in the data? And anyone else? Konstantinos, what do you think about gaps in the data? Where do you think we need to fill in some blanks here?
Yeah, I mean, the basically we have learned how we need to become better scientists. I mean, this is for sure the take-home message for my personal career and experience in general. You know, and I think it is sort of imperative that we need a large-scale randomized study for the CLTI population indication in particular. You know, building on the real-world data that seem to support safety, especially in the diseased population with a lot of comorbidities and with a CLTI indication. This is the indication that we should be looking more in the future. Building also on the best CLTI, on the best CLI study outcomes that seems to highlight an efficacy detriment of endovascular. So this is the gap. This is the efficacy gap that we need to support, and a CLTI DCB DES study, I think, is imperatively mandated. You know.
I'll go next.
Interesting.
Those are
Yeah,
great comments.
please. Go ahead.
You know, a few gaps. You know, one thing, Peter, to your comment. You know, it's interesting. You know, and some of the, like in Safe PD, the CMS data, we can track how many people get paclitaxel subsequently, and it's about 20% end up getting it over the next 3 to 4 years that didn't get it to start. It's just very hard to model that without creating biases in these studies. You know, we've talked a lot about per protocol and intention to treat, and the same idea is, you know, you can only survive long enough to get another treatment. How do you handle that? And then how much time do you contribute after treatment and when your next treatment occurs? And so there's a lot of gaps. How much dose people got, that type of stuff in the real world data is not clear. You know, to me, the stepping back though is, you know, one thing that I feel proud of from our community and what Christian did, and is how do we support safety signals and safety concerns? And this comes up every few years. We had this on the coronary side with stent thrombosis. We had the paclitaxel. We had an EVAR panel last year. And so I think continuing to generate this infrastructure for generating timely evidence for safety concerns is really a part of where this field needs to go. We need to design better randomized trials. We need to do better regulation of how trials are conducted and patients are followed. And then even when we do everything perfectly, we're going to be stuck with questions and things that come up after approval. And we really need an infrastructure to be able to address those timely and provide some sort of at least directional, you know, should we keep moving forward with this product or not? So I'm really proud of what we've done. I think some areas that we need to improve, again, are better identification of specific devices that don't rely on a registry. And so there's been a lot of talk about creating unique device identifiers so that even in claims data, you can figure out the specific device used. Also, you know, cause-specific mortality is really challenging, and better data that comes from the states in terms of identifying cause-specific death are going to be important extensions of, if we do see a signal with all-cause mortality, how do we connect it to the exposure? So, you know, there's areas to grow here, but I think that, again, a lot of good have come out of this in terms of the infrastructure we have either built or are building and redesigning how we do clinical trials.
Yeah, all good comments. My sense also is that the collaboration between the specialties has never been better. The collaboration between the industry folks that make the devices that we use has never been better. And I think we'll benefit hopefully from this long term. Christian, any closing comments?
Yeah, for me, this selection bias of trials, of recent trials, is mainly driven by inalienable excluded patients. And I find it very striking that after 20 years or even 30 years of discussion, we are still not able to include more than 20 or 25% women in these trials. I think this is one of the most important tasks for the future. We should have 50% women in all trials.
Very good. Well, with that, I'd like to thank Dr. Katsanos, Dr. Behrendt, and Dr. Secemsky. I know we all have work to do today, so it's really great that we were all able to get together. So thank you very much.
Thank you, everyone.
Ciao, see you next week.
Thank you very much.